Good morning student. Today we discuss about the next cell organelle, vacuoles. Vacuoles are membrane bound sacs. Vacuoles are membrane bound sacs. Vacuoles maintain turgidity of the cell. Vacuoles help in maintaining turgidity of cell and which maintain the proper internal balance, of cellular content. Proper internal balance of cellular content is known as the vacuoles. Vacuoles are prominently found in plant cell. Found in plant cells. 2 to 3 prominent vacuoles present in plant cells. In animal cell, they are few in number. And small. They are few in number and small in animal cells. Then the structure of vacuole. Vacuoles are bounded by a semi-permeable membrane. Known as tonoplast. Vacuoles are bounded by a semi-permeable membrane known as tonoplast. Membrane Tonoblast membrane. Membrane helps in tonoblast membrane helps in maintaining the composition of vacuolar fluid. Maintain composition of vacuolar fluid. Which is called as the cell sac, vacuolar fluid or cell sac. Composition of cell sac. Composition of cell sac differs in different cells. Composition of cell sac or vacuolar fluid is differ in different types of cells. Vacuole stores excretory products. Stores excretory products and compounds that are harmful and compounds that are harmful and unpalatable to herbivores. Stores excretory product and compounds that are harmful and unpalatable to herbivores, thereby protecting the plants. Thereby protecting the plants due to harmful and unpalatable compounds to herbivores. Attractive colors of the petals. Attractive colors of the petals are due to the storage of pigments. Due to the storage of pigment in vacuoles. Storage of pigments in a vacuoles. In some large plants cell, a single large vacuole occupies. In some large plant cells, a single large vacuole occupies the central part of the cell called central vacuole. Which occupy the large part of the cell, hence called central vacuole. In such cell vacuole can occupy about 90% of the total volume of the cell. The cell sac or vacuolar fluid. The cell sac or vacuolar fluid of central vacuole is a store various ions and is a hypertonic to cytosol. Hypertonic to cytosol. Small vacuoles in seed. Small vacuoles in seeds of certain plants. Seeds of certain plants store organic material. Stores organic material like proteins, intake of food or foreign particles by phagocytosis. Intake of food or engulf foreign particles by phagocytosis. Phagocytosis involves formation of food vacuole. Involves formation of food vacuoles. Involves formation of food vacuoles. In fresh water, unicellular forms like paramosia. In fresh water, unicellular forms like paramosia. Excretion and osmoregulation takes place by excretion and osmoregulation takes place by in paramosia, excretion and osmoregulation takes place by contractile vacuole. Phagocytosis by food vacuoles and excretion and osmoregulation takes place in paramosia by contractile vacuoles. In protists, food vacuoles are formed by engulfing the food particles. In protists, food vacuoles Vacuoles maintain the turgidity of the cells. Then the next cell organelle is the Glyxoysomes. Glyxosomes are microbodies containing enzymes. Glyxosomes are microbodies containing enzymes for oxidation of fatty acid. For oxidation of fatty acids. These membrane-bound organelle contains enzymes that convert fatty acid to sugar. Contains enzymes that convert fatty acids to sugar. Then, instead of germinating seeds, in cells of germinating seeds, in cells of germinating seeds, the cells utilize stored fats. Cells utilize stored fats. Cells utilize stored fats as a source of sugar. Source of sugar. Till it starts photosynthesizing on its own. Structure of Glyxosome shows central core, then surrounding to central core, matrix, and bounded membrane. Structure of Glyxosome. This is about the cell organelle, micro body Glyxosomes. Then next cell organelle is the mitochondria. Singularly, mitochondria. Next cell organelle, double membrane bound cell organelle, mitochondria. Mitochondria is known as the powerhouse of cell. Mitochondria is known as the powerhouse of cell. Mitochondria plays significant role in aerobic respiration. Play significant role in aerobic respiration. Significant role in aerobic respiration. Mitochondria are absent in prokaryotic cell. Mitochondria are absent in prokaryotic cell and red blood corpuscles. And RBCs. Absent in prokaryotic cells and RBCs. The structure of mitochondria shows double membrane bound structure, oval or spherical or spiral plate-like structure. Oval, spherical, double membrane, oval or spherical or spiral plate-like shape. Number is differ in different type of cells. Outer membrane. Inner membrane shows a finger-like projections. Finger-like projections or plate-like projections. Finger-like projections or plate-like projections. This finger-like enfoldings is known as the cristae. Inner membrane. Inner membrane shows enfoldings called cristae. Mitochondria is a double membrane bound organelle. Outer membrane is permeable to various metabolites. Permeable to various metabolites due to presence of porine or protein particles. Porine or protein or Parsons particles. Then inner membrane is selectively permeable to few substances. Inner membrane is selectively permeable to few substances. Outer membrane is permeable to various metabolites. Presence of porine or Parsons or protein particles. Protein particles. Inner membrane, which is a selectively permeable to few substances only. Both membranes are separated by intermembrane space. Both membranes are separated by an intermembrane space. Intermembrane space. Then inner membrane shows several enfoldings or plate-like folds called cristae, appears in numerous particles. Called as the oxysomes. Called as the oxysomes or F1 particles. Or F1 particles. Which in infold or in each infold shows a numerous particles called as the oxysomes or F1 particle and cytochromes, that is the electron carrier. Each infold bears numerous oxysomes or F1 particles and cytochrome or electron carrier. Inner membrane consists of cavity called as inner chamber. Consists of cavity which is called inner chamber. Containing a fluid matrix. Containing a fluid matrix. Matrix contains few coils of DNA, RNA and 70S ribosomes. Matrix contains few coils of DNA, RNA and 70S ribosomes. Lipids and various enzymes. Lipids and various enzymes. Matrix contains few coils of DNA, RNA, 70S type of ribosomes, lipids and various enzymes of Krebs cycle and other pathways. Of Krebs cycle and other pathways. Then structure of oxysome or F1 particle. Structure of oxysome or F1 particle. Inner membrane of mitochondria. Inner membrane of mitochondria bears a number of particles called as the oxysomes or F1 particles. Each particle consists of bulbous head, stalk and base or foot. Each particle consists of head, stalk, base or foot. Head faces towards the matrix. Head faces towards the matrix. Head faces towards the matrix and foot is embedded in inner membrane. Foot is embedded in inner membrane. Head acts as an enzyme ATP synthetase. Head acts as a enzyme Head acts as a enzyme ATP synthetase. Head act as a enzyme ATP synthetase. And food act as a and food act as a and food acts as a proton channel. Oxysomes involved in proton pumping. And ATP synthesis. Head of oxysomes which act as an enzyme ATP synthetase. Food act as a proton channel. Oxysomes involved in proton pumping and ATP synthesis. This is about the mitochondria, which is the double membrane bound cell organelle, which is also called as the powerhouse of the cell. Thank you.